So thank you for joining our September Physics Matter Colloquium. So it's now the 35th edition of this virtual scientific expedition. So traveling through the Middle East with the Sesame Project and far beyond. So this live event, an open series colloquia, is prepared by the Forum of International Physics, the FIP, at the American Physical Society. So it has been created in the framework of the program Physics for Development, and our colloquium and forum aim at sharing and engaging scientific community and any curious mind. So we are sharing knowledge across a wide spectrum of topic and multidisciplinarity, and this is what we really need in the field of science. So please visit our webpage where you can learn much more about those exciting destinations and that are opening gates thanks to renowned international scientists whose thinking is resonated with the FIP philosophy, so to learn and teach why physics matters. So this month we will learn more about physics and about medical, so the medical physics. So this is one of the pillars for our healthy society, isn't it? So let's introduce the member from our panel. So we have Luisa Cifarelli with us today, who is the former chair of the forum before me, and she's also the founder of this Physics Matter. And my name is Christine Dan, so I work at the European Spallation Source in Sweden, and I'm the current chair of the FIP. And we have now our guest of honor, so Richard Spencer, who is chief of the magnetic resonance imaging and spectroscopy section, so at the National Institute of Aging, so at the National Institute of Health, so in America, so in Baltimore, in Maryland. And his group perform biophysical and physiological studies on human subjects, experimental animals and tissues and cells preparation. So the current focus on his studies of brain and muscles in normative aging and are in the setting of specific pathology of different interest in the aging population. So the technique applied a lot of those conventional resonance magnetics imaging protocol, so to specialized mathematical approach to extract tissues information from the magnetic resonance signal. So Dr. Spencer, he's a fellow of the American Physical Society and the American College of Physicians, so not physics, but physicians. So the normal, Weiner Center for Harmonic Analysis of the Department of Mathematics, the University of Maryland College, and the American Institute of the Medical and Biological Engineering. So with all this, nice introduction, I think in terms of looking at all the different aspects as well of your research. So we will now let you the ground, and we will have possibility to answer the question later after your presentation. And we can then have you as well ask your question live as you wish. So Dr. Spencer, then the floor is yours. And you need to unmute.
Thanks very much, Christine. It's really a pleasure and a privilege, especially to have the opportunity to present some of our work here. And I hope that going through this, I can convey some of what goes on behind the scenes in developing new MRI modalities. Let me share here.
We can see your screen.
Right, I'm just pushing aside all these windows and closing them. So this is about macromolecular mapping, and I will show as the slides go on how this relates to the part about beyond free water. So as Christine said, I'm at the NIA, part of the National Institutes of Health in Baltimore, and this is work that we've been doing for the last several years. Now, somehow, all right. So the focus of this talk is going to be on something called myelin, and that may be familiar to some of you or perhaps not. It's a fatty substance that coats axons and potentiates electrical transmission in the nervous system, central nervous system and peripheral nervous system. Central nervous system means brain and spinal cord, and peripheral nerves are sort of everything else. This is just a schematic of how nerves, of the nerve structure. And you see the myelin sheath extending along this axon here. And again, electrical transmission occurs in a complicated way that I'm not going to get into. Here's a micrograph of this just from Wikipedia, and you can see this sheath here, which is very important in what I'm going to be talking about, of the lipid substance that wraps around axons. Now, you can imagine here there may be water molecules trapped in this, and in fact, that's the theme, in a sense, of this talk. Now, demyelinating diseases are very important in clinical medicine. The one that everyone has heard of is multiple sclerosis. So this is a central nervous system disease affecting the brain and spinal cord. Guillain-Barré is maybe almost as well known, and that's sort of the paradigm of demyelinating disease in the peripheral nervous system. These are studied clinically with MRI, of course, and just looking at multiple sclerosis, there are many indirect MRI markers for demyelination. Again, the demyelination interrupts conduction, electrical conduction along axons, and inhibits motor function. Later in the disease, it also inhibits thought processes. So it really can be quite devastating. And it's followed with MRI. There's a variety of MR modalities that can assess this. Magnetization transfer is one of them, looking at relaxation times, T1 and T2, and diffusion tensor imaging. I don't have time to get into all of these, but suffice it to say that this is not, this is done routinely and on a clinical basis. Now, here's a typical image just downloaded from Mayo Clinic showing some multiple sclerosis lesions and in their typical location. And it's a conventional T2 weighted image. So what's the issue here? Well, the issue is that it's not quantitative. In other words, if you measure the T2 here and it's a certain value, let's say it's 80, that doesn't mean that there's twice as much as if the T2 were 40. So there's no real quantitative relationship between the amount of myelin and the changes in, say, relaxation times or the diffusion tensor values. On the other hand, there are more direct MRI markers, which are myelin measurements that are based on multi-component relaxation time analysis. And this you'll see a lot of in the next slides. So you'll certainly know more about what I'm talking about here. This was actually developed by Alex McKay some time ago, but it's been the subject of very active research ever since that time and has become increasingly important. And this is much more specific, so that we have a marker that, for example, when it changes by a factor of 2, that means there's a change by a factor of 2 in the amount of myelin. So that's what I mean by direct and quantitative rather than sensitive, such as these indirect markers, but not quantitative. So demyelination is also a factor in dementia and aging. Now, interestingly, we don't think of demyelination as readily in that area. We think of it again as in the classic demyelinating diseases. Multiple sclerosis and Guillain-Barré are just two of many, but perhaps the most prominent. But it also occurs in dementia and aging, and we're especially concerned with that at the National Institute on Aging. Now, Alzheimer's disease in particular is a hugely expanding global problem with the aging population. And most of the research on this, prompted by Alzheimer's findings about 100 years ago, is on the so-called beta amyloid hypothesis. Again, I'm not going to get into the details of this, but even in his original paper, Alzheimer made comments about myelin. So the paper was not just about these plaque-like structures, although that's what's really been pursued, but he even made at that time very insightful comments about myelin sheaths being basically greatly and diffusely rarefied, in other words, absent or less robust. And then additional comment, in our case, the decay of sheaths in the white matter and the lateral pyramidal tracts of the spinal cord can be established. So Alzheimer even then showed a great deal of insight by highlighting not just the plaques that have been studied so much, but also the myelin sheaths themselves. So in fact, accelerated myelin loss may be a central process in the pathophysiology of mild cognitive impairment and Alzheimer's disease. This is the so-called Myelin hypothesis, popularized by George Bartzokis at UCLA, really starting about 20 years ago. He died prematurely and very sadly. And I just love this quote from his wife talking about him, talking about his creative mindset. She said he didn't even know there was a box to think out of. I love that quote. Now, what are the potential causes for this accelerated myelin loss? Well, potential causes include decreased cerebral blood flow, and that's because cerebral blood flow is essential to provide substrate and oxygen to tissue in the brain. And in fact, myelin maintenance and production is very energy intense. So decreased blood flow could certainly coexist or even lead to myelin loss. Likewise, increased iron can do this. And in fact, myelination deficits are increasingly interesting in the sense that we now think of the brain as many connected units rather than just single areas doing a single thing. This is an emerging paradigm, and myelination really potentiates this view because it, again, acts as something which enables proper transmission of electrical signals between regions of the brain. Now, this talk is on macromolecular mapping, and by that I mean myelin mapping in particular, although there are other macromolecules as well. In fact, we were studying cartilage before we started working on myelin. Now, the point I want to make here is that conventional MRI maps basically free water. And so all the different MRI modalities that you see relate somehow to properties of free water. When I say free water, I don't mean as free as if it's in a test tube, but relatively mobile water. Now, about 10 or 15 years ago, we really started to want to map macromolecules. And this really highlights a distinction between macromolecular mapping and free water mapping in MRI. Now, conventional MRI is a mature technology. If you want a beautiful MR image, you just go to your local MRI facility and conventional routines are implemented and you get a beautiful image. That's basically a mature technology. And in fact, mathematically, it's an easy math problem in a sense, and I'll explain that sense. Now, there are innumerable technical difficulties and advances, but in a certain sense, it's an easy math problem with a so-called error multiplication factor of 1. And I'm going to talk more about this. I'm talking about what will be called more technically a condition number. The point here is that when there's errors or noise in the data, that translates into errors and noise in the resulting image and parameter estimation without any amplification. So the same amount of error, roughly speaking, that is in the data is that amount of error, that same amount of error occurs in the actual images and analysis. Now, macromolecular mapping, on the other hand, not water, but macromolecular mapping, turns out to be a hard math problem, again, with the same innumerable technical difficulties, but this error multiplication factor, which really multiplies the error from the data into the product, can be 100, thousands, or even more. Now, to explain this, I have to spend one single slide on MRI that you all might not be completely familiar with. I'm going to be talking a lot about spin-spin relaxation, or T2, and I just want to show what that is briefly. This is a vertical bore MR magnet. A clinical system is horizontal bore. I'm choosing this vertical bore because it was the first magnet I ever had in my lab, so I'm fond of it. Now, the magnetic field in this case is vertical because it's a vertical magnet. This is all rotated 90 degrees for a clinical magnet. The magnetization vector in the tissue or sample in the magnet is aligned, this is Curie's law, is aligned along the main magnetic field. After an excitation pulse, which is a radio frequency pulse that tips the magnetization vector, that vector begins to precess at some number of megahertz. So in a conventional 1.5 Tesla MRI system, it precesses or rotates around the B0 axis at about 64 megahertz. 3 Tesla system, it's about 128 megahertz. So it really precesses quickly. That's interesting, but what we're really focusing on is the fact that the amplitude of that rotating vector decreases according to an exponential decay law. T is the time over which this is precessing, and T2, the spin-spin relaxation time, describes the time constant of the decay of this magnetization. So if you sample that, what does that look like? Well, it looks like a decaying exponential with this exponential form, e to the minus t over t2. Again, t2 is the time constant. And many publications, innumerable publications, have been related to the fact that this relaxation time or relaxation rate is sensitive to the biophysical status of tissue, for example, hydration. Again, I'll talk more about that. The time scale is typically tens of milliseconds. And again, amplitude on the vertical scale, time on the horizontal scale. This just shows the decay of the amplitude of the magnetization as it spins at megahertz in the magnetic field. Now, water with greater mobility, I'm calling this free water. Again, free is too strong. It's not ever free like in a test tube, but it's let's call it mobile. That relaxes at a relatively slow rate. This is because dipole-dipole interactions tend to average out so that there's less dephasing that occurs. Now, with reduced mobility, and by this, I really have in mind the water that's trapped between the myelin layers, as I illustrated in a previous slide. This reduced mobility water, in contrast, relaxes much more rapidly, again, because the dipole-dipole interactions actually are greater with less notional averaging. And that's what I mean when I talk about beyond free water in the title of this talk. We're talking really about water that has reduced mobility and relaxes rapidly, and in particular, it relaxes at a different rate than the more free water. So let's go back to the early work that when we were studying cartilage water. So cartilage, again, is the white material at the end of bones. It tends to decay with arthritis. And the goal here is to determine the parameters from the data. So we collect data and we try to determine, we want to say something about the health of the cartilage. Now, again, here I can't go into great detail. Suffice it to say that there are different water compartments. And when I say water compartments, I don't mean spatially separated. I mean that within a certain tissue area, there's some water that's associated with collagen that relaxes so fast that we can't see it actually with conventional methods. There's water associated with other macromolecules called proteoglycans, and there's the more free water. Now, the proteoglycan water is what we're interested in here. And again, that is much more bound. Proteoglycans are important molecules for cartilage function, and we want to identify what is the proteoglycan content of the tissue. Now, here I can show you one of our early results where we are looking at these tissue compartments of water before we degrade the tissue and after we degrade the tissue. This is a in vitro experiment mimicking arthritis. And what we can see is that there's a shift from the red to the blue. All the red peaks are pushed over to the right with greater T2. Remember, greater T2 indicates greater mobility. So when we degrade the tissue with this enzyme called trypsin, we move from the red, which is more bound, to the blue, which is less bound. So that indicates tissue degradation. And we want to be able to say something about that in cartilage tissue and to create maps of it. So here's a comparison. This is a conventional T2 map of cartilage. And T2 here is affected by the water content, as mentioned before, macromolecular content, collagen orientation, tissue architecture, and so forth. So it's a very sensitive marker, but it's not specific to how much proteoglycan there is. So in 2011, we developed a method for mapping this, where we look at the T2 distribution of the tissue and then separate that on a pixel by pixel basis and have a map of this very interesting macromolecule called proteoglycan, as mentioned here, that provides compressor resistance on the one hand, and separate that from the unbound or freer water. So this is a way to directly define the distribution of proteoglycan as compared with the loosely bound water. So again, this is 2011. And note that this image is of much lower quality, just visually, much lower quality than this very pretty mono-exponential conventional MR image, but it has much more information. Here we just know that the T2 changes. Well, why does it change? Maybe water content, orientation, who knows what. Whereas this is much more specific, showing the amount of proteoglycan actually in that tissue. Now, this is an example of an inverse problem, which I'll be talking quite a lot about. And that can be summarized here as determining parameters from data. So we observe something, in this case, an MR image. And from that, we want to get tissue properties out. This can be T2 or T1 or diffusion coefficient. T1 in the rotating frame, metallization transfer, and so forth. But we want to get parameters from data. This is, of course, a universal problem, not just in MRI. This is our version of it. Now, when I talk about an inverse problem, the question is, well, what's the forward problem then? I mean, inverse must be the inverse of something. And it's the inverse of the situation where if you were to have parameters, you can synthesize the data that you would get from those parameters. This is quite easy. If you know what these parameters are and you know what your imaging sequence is, you can readily synthesize an image. That's not hard. What's hard is starting with an actual experimental image and doing the inverse problem, solving the inverse problem, and getting parameters back. I'll just mention, if you want to read more about this, there's a well-known review article in Review of Scientific Instruments in 1999, which is really very worth reading. And then we wrote a review article more recently, which covers much of this material on inverse problems in magnetic resonance relaxometry. Now, inverse problems, again, are determining parameters from data. Going back to the literature, the first mention I could find of this is from Ovid about 2,000 years ago. There may be something earlier, but I don't know what it is. And he very insightfully talking about human condition, saying the cause is hidden, but the result is known. And that is exactly an inverse problem. We know the result. How can we get the cause? Well, a little bit later, Tikhonov came around. And of course, others studied this before, between Ovid and Tikhonov, but we'll be talking about Tikhonov's regularization in particular. So Tikhonov developed a method along with others, but his name is associated with this, developed a method to stabilize or regularize these inverse problems. I mentioned that these problems are very hard to solve, and one reason is because they're so sensitive to noise, as I'll illustrate. Whenever I use the word regularization, you can think of stabilization, meaning that with different noise realizations, you would get similar results for a regularized or stabilized problem. But if you don't regularize or stabilize it, you get very widely differing results from different noise realizations. So how does this, what is the general framework for this? Well, in general, you all are very well aware that Fourier analysis is extremely powerful and useful for separating frequencies from a time signal. So here's a time signal that in this time signal, there's a superposition of sine waves, basically, at different frequencies. It looks like a mess, but as you all know, you can dissect this very, very nicely with Fourier analysis into a frequency spectrum. We do this intuitively. We don't even think about it anymore. It works. We love it. And we don't think about the complexity of it in a sense, because in some sense, it's not complicated. It's a very stable procedure. Small errors in the data remain small in the frequency spectrum. However, different water pools, meaning, again, not physically separated, but water pools that have different water mobility, they have the same MRI frequencies, but they have different relaxation times, as I indicated previously. So Fourier analysis won't work. You'll get one peak at the water frequency, and that is not going to help you distinguish among these different water pools. So instead, we have to distinguish them based on relaxation times or diffusion characteristics, basically some sort of exponential decay differences. So this is a special case of the Laplace transform, or the inverse Laplace transform, when you are trying to dissect the superposition of time constants. If you can correctly perform the inverse Laplace transform, then you can get a spectrum. I don't like that term so much. It's used, however. You can get a spectrum, or better yet, perhaps a distribution of relaxation times. I mentioned that the tissue may have different relaxation times for more bound water versus less bound water. However, what you observe is simply some decay. You want to separate those. The way to do that is with an inverse Laplace transform, and then you would get this distribution of the different relaxation times in the sample. This all looks very analogous to the Fourier transform, or inverse Fourier transform. The inverse Fourier transform and the Fourier transform behave identically mathematically. The problem is the inverse Laplace transform is very badly behaved, and small errors in the data propagate into very large errors in the relaxation time distribution. We can illustrate that by something called the condition number. A condition number is a description of how close a matrix is to being badly behaved, or how badly behaved the matrix is in terms of inversion. It's a little more, well, that's the basic sense of it, although inversion, this also accepts non-square matrices. So there's other criteria, but condition number basically tells you how badly behaved the matrix is and how redundant the columns of that matrix are with each other in a least squares problem. So the formalism is written here. So zeta of K is a matrix, zeta is its condition number, and there's some error in the data, and that the degree of that error is multiplied by the condition number of the matrix to propagate into the amount of model error. So again, in the Fourier case, we see this was a small number. In the Laplace case, this was a big number. So let's look at the discretized Laplace transform. Now, this, of course, is just one example of a discretized Laplace transform, which we want to invert to get parameters from data. And this has a certain number of columns and rows, so that the actual values for the condition number are dependent on the details of this matrix. But in general, you can say that this condition number, which again multiplies errors into your resulting error, can be huge. It can be 100, 1,000, 10 to the 5th, 10 to the 10th. As the matrix gets bigger and bigger, oh my goodness, this condition number far exceeds machine ability to handle these values. So this is a pretty upsetting situation, and it's the one that we're left with. Now, the Fourier transform, on the other hand, this is again just a certain manifestation of a Fourier transform matrix. This is an orthogonal, or really Hermitian transformation, and in that sense, it has the optimal condition number of 1. No matter how big it is, the condition number is always 1. So looking at this, you can see why the world of the Fourier transform is so much simpler to deal with than the world of the Laplace transform. And as mentioned, this is the realm of conventional MR imaging, which is why I said that it's a relatively easy math problem. So MR imaging, conventional MR imaging, the mature technology, it's a Fourier technique. Data is collected in something called K-space, which is the Fourier transform of the image that you want. This is just a property of how the imaging data is collected. It has to do ultimately with the precession of magnetization in the magnetic field. But briefly, once you have the data set, you take the inverse Fourier transform. Again, that behaves the same as the forward Fourier transform, and you get your visualized data. This is stable. Now, unfortunately, the Laplace transform, as mentioned, is what we need, but it is not so well behaved. It comes up frequently, though. T2 decay, T1 decay, diffusion signal attenuation. It's very easy to derive mono-exponential decay parameters from a single curve, a single component. But once you start adding up these components, it's very hard to tease them out. Again, if these were different frequencies, Fourier analysis would allow you to do that very neatly, but Laplace analysis does not allow this in any easy way. It's an intrinsically ill-conditioned problem. The whole idea of badly conditioned problems was formalized by Hadamard 100 years ago, and the couple of the criteria that he laid out were that the solution to an ill-posed problem is very sensitive to noise. In other words, one noise realization versus another noise realization may give you wildly different answers. But of course, as a scientist, you don't know whether you have this answer, whether this answer is correct or the other answer is correct. And similarly, there are multiple solutions to a given data set that are nearly equivalent in terms of the data, but look very different in terms of parameters. Now, these two are closely related. A simple example, perhaps the most classic example, is multi-exponential decay. When capital N is 1, it's easy. It's a mono-exponential. But when N is 2, even 2 becomes hard, depending on the separation of time constants. But if this is the signal, S of t, and you want to extract from that constants in the degree of the size of the components and the relaxation times, in other words, to derive this set of parameters, that's an ill-posed problem. Now, in 2015, we developed and implemented a Bayesian stabilization technique to address this problem. You can never say solve this problem, but you can address it. And this was based on some very important work of Neil and Bretthorst from about 30 years ago in a really nice paper that I think is really a classic. So the Bayesian technique, you've all heard of Bayesian analysis, and I'm going to just show some pictures of that. So we want tissue properties, as mentioned, but what we have is data. And remember, going from this data to these tissue properties is an ill-posed inverse problem. And it's hard in that sense. On the other hand, if you are given these tissue parameters, as mentioned, it's not hard to go and synthesize an image. So that's a much easier forward problem. Now, Bayesian analysis also has prior knowledge or assumptions, which can either be a bug or a feature, as they say. It certainly elicits a lot of controversy. We think of it much more as a feature than as a bug. But in any event, it does incorporate prior knowledge or prior assumptions. So this is Bayes' theorem. And the way I think of this is that it converts a hard inverse problem into a much easier forward problem. And then the tissue properties that we want, namely these parameters, are found by maximizing the probability of acquiring a given data set. So this is very related to maximum likelihood estimation, although it's not quite the same. Now, using this analysis method, which stabilizes this inverse problem, we returned a few years later to the cartilage problem. And I'm just showing this slightly out of sequence because I introduced the cartilage problem, and it really illustrates the Bayesian method before I move into brain. So again, this was our first proteoglycan map, 2011. And now, using this method, we can create in vivo maps in less than half an hour in, again, living subjects that are much higher quality. And here we're looking at the femur and the tibia, and cartilage is the colored material in between them. As mentioned, it's the stuff at the end of bones that goes bad in arthritis. And you can see that from the color bar that there's a variation in the amount of proteoglycan in this tissue. And that's exactly what we're trying to map. Now, you might say, I don't know, this looks very, very jagged. It looks like there's a lot of specks here. Is that noise? It looks like noise to me. But one way to check that easily is to take two noise realizations. In other words, perform the experiment twice, one after the other. Obviously, the noise realizations are different in that case. And doing that, we can compare and see whether these non-uniformities are intrinsic to the tissue, in which case they're proteoglycan, or if they're really just a property of the noise. So here are proteoglycan maps from two identical acquisitions, one following the other. And interestingly, these distributions are almost identical in these two completely independent images. So we are pretty confident that this reflects the tissue proteoglycan distribution rather than the properties of noise. I won't show this, but doing this with conventional methods like nonlinear least squares leads to wildly different patterns. Now, the rest of this is going to focus on brain. I just wanted to show how powerful this Bayesian method was by illustrating it on cartilage, where we had pre-Bayesian methods as well. Now, here's our first application for myelin water fraction. Now, when I say myelin water fraction, you can think myelin. The reason I'm calling it myelin water fraction, or MWF, is because it's really water that is trapped within the myelin. So it's the amount of water, it's the proportion of water that's trapped in the myelin sheaths versus the freer water. So here's a high signal to noise standard image, standard or fiducial image. And this is acquired under conditions and filtered under conditions that are not necessarily clinically feasible, but nevertheless, they represent a gold standard. Of course, without simulations, you don't know the right answer, but we're doing as well as we can here with very high quality images. Now, conventional analysis for a low signal to noise image with nonlinear least squares, so NLLS is nonlinear least squares, conventional method, there's certainly a nice relationship between these images and the fiducial or gold standard images, but you can see there's a lot of variation. Now, the same data, but processing this with the Bayesian analysis that I mentioned and illustrated, you see much better results. There's a much greater similarity between these Bayesian results versus the nonlinear least squares results as compared when compared with fiducial image. And of course, we can quantify this as we've done extensively, but just to show this visually, we can look at these error maps and you can see that the errors are considerably greater in the nonlinear least squares conventional analysis as compared to the Bayesian analysis. These just show difference images, and basically the mean square error throughout that image. And the mean square errors are considerably lower in the Bayesian analysis. Now, since that time, we've moved on to a different signal model, which is not the bioexponential, but rather one that's experimentally very efficient, although numerically quite difficult. And the details of this are unimportant. I just wanted to show the model. This McDespot model developed by Dione and presented in 2008 was really an important advance and allows us to collect data from which we can extract relaxation times using steady state, so-called MR steady state sequences. These are the signal models, the SPGR, spoiled gradient recalled echo signal, and then the balanced steady state free precession signal. Again, the details of this are not what's important, but that's the model that we use. Now, how does this work out? Well, we collect McDespot data. That's this method I just showed on the previous slide with the more complicated matrix analysis, a matrix framework, and treated that with nonlinear least squares. Now, and here's some abbreviations here: signal to noise, Myelin water fraction, which is what we're after, nonlinear least squares. Now, why does nonlinear least squares fail so readily? And this is true not just for Myelin water fraction mapping, but really throughout a lot of applications of nonlinear least squares. You end up with flat energy surfaces. In other words, and this is real data, by the way, this is not an artist's conception. This is the least squares residual. This is the Myelin water fraction. And you can see that there's a very shallow minimum. The least squares algorithm wants to find the lowest value here and declare that the correct Myelin water fraction. And there is going to be a lowest value. But that lowest value, as you can imagine, will change between noise realizations because these are also so similar that small variations in the data will lead to large variations in the optimal value or the minimum value on the x-axis. So for this noise realization, say the value is 0.15. Some other noise realization could be 0.3. So which number did you publish? It can be just very noise realization dependent, and that's going to lead to a lot of instability and these very irregular looking images. Now, the Bayesian analysis of the McDespot signal, again, this is not bioexponential, but you can think of it as just another hard inverse problem. The Bayesian analysis ends up showing images as seen here, and these are much more uniform. Again, quantifying these errors is something that we do extensively in the papers, but there's no right answer in a brain because you really know roughly what the values should be, but you don't, of course, have the correct answer in front of you because these are not simulations. Now, how does this do better? Well, in Bayesian analysis, you don't recover a certain value the way you do in nonlinear least squares. The algorithm doesn't say, oh, the number is 0.15. Instead, it gives you a probability distribution function, or PDF, of the value of the parameter as a random variable. This is something that people object to in Bayesian analysis. I don't think it's a great objection. I think it's fine because you have uncertainties in nonlinear least squares as well. But anyway, that's a feature of the Bayesian analysis. Again, feature or bug, we think of it as a feature. So here's the probability distribution function on the vertical axis, Myelin water fraction on the horizontal axis. And from this, you can extract your Myelin water fraction parameter. We tend to use the maximum posterior estimate, namely the peak of the PDF, but other ways can be done, can be implemented as well. For example, calculating the first moment. There was a nice perspective on this published in Science when we were sort of in the middle of this work, and this talks a little bit about perspectives on this work and comments from other experts in the field. Now, I've shown in sort of a very brief form the basis of this method, and now I'm just going to show a couple of our many applications. I'm not going to show all of these, but now that we can map myelin in what we believe is a fairly accurate fashion, we can relate that to a lot of very interesting and important criteria, or I should say phenomena in brain aging, and brain pathophysiology. Now, we're very, very lucky to have great access to the Baltimore Longitudinal Study on Aging, which is it's really one of the world's longest running studies of aging. This enrolls volunteers 20 years and older, and just follows them until death, basically, or until they drop out. But these are motivated subjects, and they tend to stick with it, and provide and take a wide range of tests, ranging from conventional blood tests and blood pressure, height, weight, things like that, but all the way to very advanced genetic markers, imaging methods, really molecular markers, just almost anything you can think of. And this is a vast database that we can then mine for relationships with the myelin water fraction maps that we're creating. I'm going to show a couple examples and then move on to some methodology. So again, using the method already shown, this looks at cerebral myelination patterns in normal aging. The vertical rows are different age groups. These are all registered to a common atlas so that they can be superimposed. And the myelin water fraction is shown on the vertical, on this color bar here. So you can see in the middle of the brain, which is where white matter is. White matter is myelinated. Gray matter is not myelinated. That's on the periphery. So again, just to be clear for this, white matter is where we expect myelin. It's white because of the myelin. That's on the interior of the brain. Gray matter is the more cellular part of the brain, which tends to be located in the periphery of the brain. So we see the number of subjects in each age bin from 20 to 29 at the lowest, youngest age, up to 90 to 99 years old in the highest age group. And we can see the myelination patterns visually. Now, the horizontal rows are just different slices through the brain. So you can pick your favorite row, really just concentrate on that. My favorite row is slice 2. You can see how the myelination is very well established at age 20 to 29. Seems like it's going up because it's getting redder. Then it seems like it's going down. And in fact, analysis of this shows exactly that trend, where there's a little bit less myelin at the younger end of that age group. We don't have data at this point on individuals even younger than 20. But this myelination pattern does tend to increase up to say middle age and then decline with more advanced age. And again, this is normative aging. This is not individuals with any impairment. Now there's whole brain data, and there's also data for many identified brain regions illustrated here. The point is that we can actually use this method to get general trends in myelination in the brain. Now, of course, there's a lot of scatter in this. These are not laboratory animals that are all clones of each other or very similarly related. These are very much related. These are human beings with all the variations of genetic background, lifestyle, everything you can name. So, of course, there's a lot of variation. This is pretty typical for physiological data on humans. You don't get nice physics curves, which can be quite distressing, but that's what you get. Now, this shows how higher function really requires development of higher function sort of co-occurs with greater myelination. Now, in the brainstem actually is a little bit different. That's lower part of the brain where this is required not just for thought, but really for basic functions, breathing, heartbeat, attention, posture, things like this. And so that has to develop relatively quickly. So we can do the same thing here in the brain stem and look at myelination patterns. Again, the vertical columns are superimposed scans over age decades, and the horizontal rows are different slices. Pick your favorite row. Now we can quantify that as well. And here we see a somewhat different pattern, which is basically what we hypothesized. And here we really don't see this kind of curvilinear relationship, but rather more of a linear relationship trending downward. Now, those of you that are experienced in curve fitting are well aware that you could certainly fit a higher order curve. Obviously, we do statistical tests to decide the number of fit parameters we'll be using. On the previous slide with these parabolic shaped relationships, that's not mathematically required as a parabola. You could fit many different shapes to it, but the point is that there is a different pattern of overall monotonic decrease in most parts of the brainstem versus this inverted U shape found by others using less direct methods in the actual brain. Now, let's move on to pathophysiology. Again, myelin loss, the involvement of myelin and myelin loss in MCI and Alzheimer's disease really has been put forward and achieved a lot of prominence by the work of George Bartzoukis, who I think did the community a huge amount of, really a huge service in really pushing this. So we studied this using our direct stabilized method, and we did find a decrease in myelin water fraction in many brain regions indicated by arrows, and really found that the extent of the myelin loss is higher in Alzheimer's disease as compared to MCI. And of course, we're amplifying these findings with much greater population. And we also found in this study that the myelin content, as consistent with the previous work I just showed, decreases with old age. So those are some applications. And I want to move on to a different phase of the talk now. This is our prime focus, myelination patterns in the brain. We've developed several different methods for stabilizing this kind of inverse problem. Now, I've showed you the Bayesian method and many results from that. One method that we have, relaxometry and higher dimensional analysis, I'm not going to show, but I'm going to try to touch on these other four, obviously quite briefly, just to show that this is not a solved problem. There's a lot of interesting applied math that can still go into this. So, as mentioned, multi-exponential analysis is an inverse problem central to myelin mapping. And the version of this I'm going to show right now has to do with recovery of the T2 distribution, not discrete values in a sum, but the distribution as illustrated previously for cartilage. So here's a multi-echo MRI sequence. It obeys this T2 decay, and a numerical inverse Laplace transform gives you this relaxation time distribution. So this is a distribution of these values are relaxation times. The vertical axis is the amplitude. So you can really see a histogram essentially of the T2 relaxation times in the tissue. Now the myelin associated water, that is the water that's less mobile, trapped within the myelin sheath, is represented by the more rapidly decaying, in other words, smaller time constant or smaller T2 portion of this. However, deriving this is very difficult. It looks simple when you just draw icons like this, but in fact, there's a lot of instability, and this is one of the things that our group and other groups are addressing. So how do we stabilize this? Well, the math problem involved in this is that of a Fredholm equation of the first kind, which I can't get into here, but the discretization of that looks like this conventional linear least squares problem. It's linear because K, which is the kernel matrix, is this Laplace matrix that I showed before when I was illustrating these massive condition numbers. M is a set of parameters that describes this. In other words, it's really the histogram values as you move across this T2 axis. So M is the desired quantity and D is the data. So this is a very classic setup for linear least squares. The problem is there's many values on this x-axis. It's a big matrix, and this thing is terribly ill-conditioned. So it's very unstable. One set of noisy data with the exact same actual underlying tissue will give wildly different values for this vector M than a different noise realization. So we stabilize that using the Tikhonov method by regularizing and adding an additional term with a regularization parameter lambda that has to be tuned, and this is very difficult to choose an optimal lambda, times the magnitude of this vector. There are many books and PhD theses written on this. I'm not going to go into details here, but suffice it to say that adding this extra term allows you to stabilize this inversion and the results for obtaining M. It allows you to stabilize that to a degree dependent on how large or small lambda is. When lambda is small, you recover the original problem. When lambda is large, you end up with a very biased and useless result, but it's quite stable. If lambda were infinite, for example, you'd end up with M equals 0. Not very useful, but very stable. So how do you adjust lambda? There's many ways of doing this. None of them's easy. So conventionally, what you do is, and this has been done for 100 years, you check the solutions, in other words, the values of M sub lambda for many different values of lambda. You choose the best result according to one of the several available criteria. This can be the L curve or generalized cross-validation or the S curve or the discrepancy principle. You choose one that's the best, you think, and just discard all the others. Well, in fact, there's information in these others. And so we developed a method called spanner regularization, which does not discard all of the other solutions. In fact, it keeps the other solutions, even the ones that are not optimal. Now, why does this work? Well, we noticed that two different solutions. Here are two different bioexponential signals with very different parameters. I even kept the coefficients the same, just the exponential time constants are different. But these are, you can imagine with noise, these are completely indistinguishable. This is a classic example of a Hadamard defined, in other words, as defined by the mathematician Hadamard 100 years ago, an ill-posed problem where very different answers give you pretty much the same solution. But I noticed that as you regularize, your values, obtained values can change along this regularization trajectory. So on the left, you would say, well, these are basically indistinguishable. Whereas on the right, same parameter sets, if you map something different, namely the value of parameters on the vertical axis versus the regularization parameter, you can see these trajectories are actually quite different. Now, they're not wildly different, but they are clearly distinguishable. Nobody would say these are pretty much the same curve. Unlike on the left, where, yeah, they're pretty much the same curve, especially when you introduce noise. These curves on the right are different from each other, meaning that model 1 and model 2 have different patterns. So we implemented that idea in span-reg regularization, where we combine multiple differently regularized solutions. This retains the stability of regularization, and the goal, which turned out to work, was to retain resolution and also greatly reduce the importance of picking just the right value of lambda. So in brief, this takes multiple different solutions as mentioned. So M of sub lambda 1, sub lambda 2, these are all different solutions with coefficients that need to be found in a clever way, and takes a linear combination of these. So how well does this work? Well, I don't want to go through all of this detail, but let's just look at the upper middle. We see the true distribution. These are simulation results, so we know the right answer. This black curve is the true distribution from which we generate data. We then add noise and analyze the data using either span reg or a conventional method such as the discrepancy principle. You see the discrepancy principle, the blue curve utterly fails. It doesn't even pick up two curves, whereas span reg does a much better job of picking up these two curves. And you see that in the upper right. You see that throughout this pattern of separating Gaussians. And of course, there's much more analysis in the publications, but this gives you a quick look at how effective span reg is. Now we apply this to brain, of course. Again, there's no real reference here. There's no gold standard, but we create a very high quality myelin water fraction and use that as the gold standard. Now here's span reg, high signal to noise data, compared with discrepancy principle and compared with nonlinear least squares. Visually, you can see that the span reg results are considerably more faithful to the original as compared with the discrepancy principle or non-negative least squares in this case to recover T2 distribution. This is all quantified, of course, in the publications, but this gives you a visual impression and shows the application of span reg. Now, another method we developed is called localized regularization. Now, how does this work? Well, we have a distribution that we're trying to recover, and it has many different features to it. There's sharp areas, there's broad areas, there can be anything in this distribution. And when you pick a regularization value, that applies to this entire distribution, and that's not great. That's a compromise where no region of this really gets what it wants. So the very conventional for 100 years method of choosing one value of lambda can't be good everywhere. It can be a compromise everywhere, that it's pretty good everywhere. So we went ahead and developed a method where we implement a localized regularization across a distribution. Now here's the conventional regularized non-negative least squares problem. I used the symbol M in the previous graph. These are P's for parameters. And again, we're maximizing, we're taking the arg min, we're maximizing likelihood, taking the minimum of this expression. But we can actually move this lambda in as a vector. So we now have a vector of lambda values multiplying this parameter vector. So that gives you a localized regularization for lambda. Well, how does this work? How can this be done? Well, it turns out that we can derive from this the vectorized version of the so-called normal equations for least squares analysis and solve this iteratively for P and for the vector lambda. So vector lambda is what we want to obtain, and from that we can get the vector P, which gives us a much nicer distribution. So this is an iterative procedure. How does this perform? Well, it performs quite nicely. And again, without going through all the details, we see that localized regularization is much more faithful to the true distribution given in black than the discrepancy principle, generalized cross-validation, or the L curve. So these are just a couple of illustrations for model systems from which we, again, derive data, add noise, invert using these methods, and try to recover the distribution. And again, localized regularization, this vectorized iterative process, performs quite nicely. Now, we also get something quite interesting, namely the actual vector of lambda values. Here's the T2 distribution, again, on the horizontal axis. This is a lambda value. Conventional methods, there's a single lambda for every single T2 value. There's just one lambda involved. We, however, obtain a vector of lambdas, and we see that this vector of lambdas has a lot to do with this probability distribution function of the actual T2 values. Again, we apply this to brain, and we can perform myelin mapping with this. Ground truth, this is, again, there's no real ground truth or gold standard, but nevertheless, this is a high quality image. Myelin water fraction determined using filtering and our best methods. And then here's the least squares, conventional method, discrepancy principle, regularized version with somewhat better performance, and then localized regularization with even better overall error. Now, I think this would be a time to just move to the conclusions. There's a couple more methods ideally I would like to show, but I think time is upon us. So I just want to kind of move to some further developments and conclusions. This is all developed for Gaussian noise and primarily with multi-exponential relaxation, but there are different noise models. Ricean noise and chi-squared noise models are extremely important in imaging, in MR imaging. This is a kind of an interesting area where you typically see, oh, Gaussian noise, we can assume everywhere. Not so much in magnetic resonance imaging. There are also many different signal models. We've looked at bioexponential primarily, but there are others. I illustrated this with McDespot, but there's also stretch exponential, kurtosis models, combinations of the above, and also diffusion analysis and diffusion and kurtosis tensor analysis. There are also spatial priors that we're developing and different penalty models. We've implemented Bayesian information criteria to determine when to apply regularization. And I mentioned quite a bit the problems in deriving lambdas, and we're developing neural network methods for actually selecting regularization parameters. This is an early result here where we see that oracle methods and neural network predictions of lambda are fairly in concert. So this is a development in that area. Horizontal axis here is lambda, and this is the histogram of values, and this is a distribution obtained over a large training set. Conclusions, there's 2 slides. First one, we're just, what I hope to illustrate is that analysis of myelin water fraction in the central nervous system is an ill-posed inverse problem. We've developed a Bayesian method that's highly stabilized for mapping myelin. We've had the pleasure of working with the Baltimore longitudinal study on aging data to form widespread and important correlates of myelin maps with other physiologic and pathophysiologic parameters, and have been able to correlate myelination status with cognitive impairment and dementia, and then items I didn't show, correlating myelination with cerebral blood flow and other functional and metabolic outcomes. Here's some publications on these areas, And the final conclusion slide, the inverse problems require mathematical stabilization. We developed the Bayesian analysis as mentioned, there's general regularization, and then there's several other types of regularization which we've been developing and trying to push this field forward so that there's the availability of multiple advanced myelin mapping techniques, highly stabilized with high fidelity, high resolution, that is high spectral resolution, or high resolution in determining T2 distributions, but also great stability with respect to noise, which is a key problem Some of this is published, some of this is in progress. And then finally, I want to just mention an outstanding group of collaborators. Mustafa Berhara, one of our brilliant scientists who did so much of this myelin water fraction mapping with McDespot. Chuan Bai, a mathematician at University of Maryland who has worked on SpanReg and LocReg. Raghu Baland, Wojtek Shaza, Pak-Wing Fok, Yvonne O, at outside universities. These are all math faculty. Luigi Frucci, Susan Resnick at the NIA, who have collaborated with us on all this brain work, and then individuals in my group, trainees who are just outstanding scientists who are really developing a lot of these methods. And with that, I will conclude, and I will thank you for your attention.
Thank you very much, so Professor Spencer, for this very complete, maybe a bit different from what we have typically on this platform, but very interesting to understand how potentially one day we could understand a bit more why we're aging. But I think that mathematically, so that's one really good link as well, connecting us. So first, do we have any question from the audience? So if we don't have a question in the Q&A, so maybe while we let some time for you to think a bit more, what do you think? So Louisa, or I had also a few questions looking maybe, so maybe Louisa first, yeah. Sorry, go ahead.
So very, very interesting. I have learned a lot of things I did not know. Somehow this myelination, which decreases with age, which is so much linked, its losses are so much linked to Alzheimer, which is one of the diseases we still don't understand. So it's extremely fascinating. But being an experimentalist from the very beginning, I had a question. How much your analysis can improve from the experimental conditions? I mean, going higher and higher in magnetic field, for instance, in the...
Yeah, I think that's a very important question. And the answer is that going to higher field will decrease errors in the data. In these relaxometry.
Sorry, but I missed, what was the field in the data you have shown?
3 Tesla.
3 Tesla.
Yeah, and I apologize.
I know of MRIs that are working at 5 Tesla, for instance, but from an experimental point of view, probably not clinically spread all over the hospitals.
Well, clinically, 3 Tesla is sort of the standard in the United States and Europe. Of course, 1.5 Tesla images, clinical images, are also beautiful and certainly very, very important. But 3 Tesla is the standard. The next step up really has been 7 Tesla whole body systems, and we're actually obtaining one of these early next year. And in general, the quality of the data, you can think of it as scaling essentially with the field strength. And that's very, very rough. But essentially, if you have, it's some almost linear function of field strength. Please, that's not quantitative, but that's kind of maybe one way to think about it. And that allows you to have smaller pixels. When you have better signal to noise, you can have smaller pixels or more rapid acquisitions. The problem here is that these inverse problems are not going to be solved by going to higher field, because these condition numbers are so terrible that, of course, we always want to have higher quality data. Another way to do this besides going to higher field is longer periods of signal averaging. And of course, for a human subject, that becomes rapidly very difficult to do. But overall, although higher magnetic field can help, it can't solve these inverse problems because you have condition numbers that are so large. So we will, as all groups, we will always combine the best possible data with the highest possible signal to noise, but that will in no way replace the need for these more sophisticated mathematical approaches.
I see. And I have a related question. Is there the same problem of myelination related to Parkinson's disease, or it's a total different?
No, that is very much an emerging hypothesis for Parkinson's disease. And to summarize this very briefly, there's definitely myelination loss in individuals with Parkinson's disease. The question is whether that's central to the pathophysiology or an associated finding. So that's a topic of current research.
Thank you.
But I do think that demyelination will be found to be a hallmark of many more diseases than currently. The list is probably about a dozen right now, but I think it's going to end up being a longer list. Where
probably not enough investigated so far because of a lack of tools. Both
I think it is a hard analysis to do, especially because it's so difficult to get quantitative data.
I meant analysis tools, of course, not.
Yeah. Yes, exactly. The analysis tools are emerging. However, it is true that conventional imaging, like T2 weighted images, are very sensitive to demyelination. It's just that you can't as readily determine whether this is really demyelination or maybe it's just local inflammation, and then inflammation will resolve, leaving you with a pattern of full myelination in that area. So multiple sclerosis, for example, has both demyelination and quite a bit of inflammation as well. And you can't really tease those apart with conventional methods. So you're right, it's hard to quantify, but it's not like there are no tools available. Quantitative tools, however, are much more difficult to come by.
Thank you.
Michael. So, and we have a question also from the audience asking also how close. So is your lab to having, so those approaches widely incorporated into the clinical use?
I would say we're not there yet. Medicine, there's a couple reasons for that. One of them is, the easiest reason to say is because medicine is incredibly conservative. And as a practicing physician, I really understand that. You really don't want to be an early adopter in general. And you don't want your physician to be an early adopter. You really want some tried and true approaches. Having said that, I think that the myelin quantification that we're embarking on is very important in terms of scientific investigation and disease pathophysiology. As far as following multiple sclerosis, for example, in other words, assessing it over time, we have yet to show, simply because it's a new area, we have yet to show that there's real advantages in, say, therapeutic dosing using these quantitative methods as compared with conventional T2 weighted imaging. There's clearly advantages in these advanced methods in terms of quantification of myelin. And that's an important advance. But I don't know that we can at this point say that there's an advantage in terms of clinical applicability.
Okay, so more calculation and more capacity as well. So indeed, you were speaking about the convergence, the resolution, so the signal. So are there any way potentially of, more data maybe with machine learning or having possibility as well to integrate such type of tools that could help you also?
Yes, absolutely. We and other groups are using machine learning. I know Mustafa Bahara at our institute is using machine learning for rapid myelin mapping. We are using neural networks for this analysis. That's one of the modalities that I didn't have time to show. And the neural network is trained on multi-exponential data or variations on multi-exponential data. So like everywhere else, machine learning is very powerful, and we're certainly implementing it. There's always the danger of introducing unknown bias. There's a lot of pitfalls, as you well know, with machine learning, but it's a very powerful general set of tools, and we and other groups are certainly implementing related methods. Absolutely.
So it means which type of capacity do you use? So high performance computing as well, or is it only, and how distributed is it? Is it in Baltimore only, or are you connected with other, maybe also university that develop such type of process? And are there other angle as well? Because it's quite often also multi-physics aspect. So are there another part as well of the faculty complementing as well those results?
Well, as far as the computing, we use cloud computing, which is a distributed set of a high number of GPUs at the National Institute on Aging computing cluster. Our collaborators at University of Maryland are using their high-performance computing resources. So some of these problems, as you point out, really require very high computing capacity. So we're making use of some of the local resources towards that, and that is actually required for some of these analyses. And I think the second part of your comment related to other physics collaborators, and I would say that we work very closely with. So creating something from magnetic resonance imaging starts with a patient or a subject and technologist and acquisition parameters, which is really the province of magnetic resonance physicists, to data acquisition, pulse sequence implementation and data acquisition, data collection, analysis. So there's a whole, I would say, group of individuals at all steps along the way here that we work with to ultimately provide or produce these results.
Very good. And you were showing as well, I think this is the important part, to benchmark also this model by tracking as well on different individuals as well at different age. So that's as well the way, I mean, this one method as well could come one day as well in a clinical aspect. So did you notice maybe some deviation, large deviation? Because that's depend a lot on maybe other condition. That's the thing. So
when you say deviation, you mean deviation among different subjects?
Exactly, among subjects that could be reflected as I don't know, I mean, something different than what normal evolution in terms of aging would be.
Yes, we definitely have, we definitely expect there to be quite a bit of individual variation in these trajectories. And we haven't developed methodology to look at variations in trajectories because we don't have a lot of repeated measurements for these myelin water fraction measurements. The BLSA study itself is decades old, but of course these myelin water fraction mappings at high field are really much more recent. So we're still developing the longitudinal nature of the database. Just in order to answer that kind of question, I think you're absolutely correct. When we're doing cross-sectional groups, we can make interesting, we can draw some good conclusions about linearity or maybe inverted U-shape for myelination trajectory, but we really don't pick up inter-individual differences, and that could be quite important for a given person to towards a prognosis for disease or development.
Indeed, to as well understand the disease and the phenomenology as well of the disease, how and what happened when. And now this is mainly indeed the dementia and Alzheimer. So I think it's clearly what it focused. Could it apply as well to any other? I mean, not only the brain, but maybe, yeah, so something else in terms of, I mean, I don't know, was it maybe even in other field, some way to think in terms of still life science applied with this method? Could you apply this method to, or were you already inquire on something different in terms of application?
Well, no, I think that's a very important question, and one area that this has been applied in, and we've worked in it for some time as well, is cartilage, where we're looking at arthritis. So mapping proteoglycan has been something that's been of great interest in particular osteoarthritis, and we did work in that area for some time, and basically I became very interested in the brain, partly because the BLSA database is so amenable to that kind of analysis, and also because of the really central importance of Alzheimer's disease and cognitive impairment in the aging population. So we emphasize brain studies and central nervous system studies, spinal cord, but certainly cartilage and arthritis is an area that we worked in. I showed some results, and is a very ripe area, I think, for further advances using this kind of inverse problem framework for macromolecular mapping.
Thanks a lot. Thanks a lot. I hope that the convergence will be fast as well, and this lambda will be powerful. So do we think about any other question, Luisa? What do you think? Or should we? let everyone.
I think that the impression is that the methods plural are extremely appealing, and you will have to collect more data, and this will help surely, because I think it's a crucial point in your analysis also to come back to anomalies in case you want to see differences in various aspects. So it's extremely interesting. Again, we need a lot of supercomputers, supercalculus, and it boils down in all disciplines that we need a lot of computer power to better solve our problems, which is great. I mean, it's a real progress the way it should be.
Yeah, and I just, as for both your comment just now, Louisa, and also Christine's comment, I'll mention in terms of general applicability, the methods that we're developing, of which I showed only a couple, are really much more widely applicable than just to biomedical imaging. The field, as you know, of inverse problems is a vast field. Yes. In geophysics and astrophysics, really throughout the physical sciences. And these methods are not, we're motivated, and our application is to human physiology and the nervous system. But certainly bioexponential analysis, other inverse problems, parameter estimation have been around for hundreds of years and recognized well before anyone thought about MRI or myelin mapping or anything I just talked about. So these are general methods as well, which we find very satisfying, of course.
Yes, but the interest is that you apply them to this particular case, which is appealing.
Yeah.
I found some of the mathematics we use in LHC experiments here, but of course it's a different context, and it's very interesting.
Yeah. And this is exactly what I was looking at as well. All the optimization function, I mean, the convergence, I mean, whether in space or in time as well, to try to understand how that could be helped. One of the things we're looking at as well for LHC or else is how actually this machine learning could help, and we can have an automatism as well in that sense. So, so that's the thing, is that depending on the different domain, sorry for the beam, so that could be really interesting to cross-correlate as well, the different type of data, depending on the application. And just as you said, so because you do the same for a given application, you cross-correlate with a different method. So that's the two aspects, the method and then the application, that are interesting. So,
we just do we're interested really in developing these methods of general applicability, but we have to have an application, and our application, to make it really interesting, is brain myelination, but it's much more widely applicable. These methods are much more widely applicable than just to that area.
Yes, but studying the brain is the thing to do. Absolutely.
Black box. Yeah.
Okay.
Yeah, and once we will be able to understand it better, maybe we can, I don't know, control. There's already a tick behind that, so hopefully, so it will be for the best in any case.
That's the hope.
So, yeah, we all hope, I think. So thanks a lot for your time. So that was really impressive to see as well how mathematics and how physics as well can go beyond the frontier to try to apply it to I mean, the health aspect. So that's something that from research will become one day as well some possibility for clinical proof as well that will complement all of that, and potentially as well more collaboration. So I think that's one of the aspects as well of this physics matter. While looking at that, I mean, right now I'm in Africa, for instance. So, and I know, and I was watching in this conference a lot of, I mean, capacity as well from the computational aspect, but from the biophysics aspect. So I'm sure there are a lot of possibility. So thanks again, so Professor Spencer, and so I will then just finish by sharing my screen and then showing. So for the next months, we will have this time. So the possibility to look a bit more also in the DNA, but of dinosaurs. So it will be looking also in terms of, and you will see it's fascinating how, so Professor Phil Manning, so from Liverpool will explain, Manchester will explain this physics looking at imaging also of life in depth, and how we will have an idea as well of what the dinosaur did, and how also with life science, but applied to and with light source, we can best understand what life was before. And you can maybe not see it well, but here this is a part of the remain of a dinosaur, of a dinosaur that you can see there. So let's stay tuned. And the 26th of then October, so we'll have possibility to learn more about that. And so in the meantime, so I think that we will have the recording hopefully available next week. And so once again, so we thank all of you for your time, and we'll hope to see you soon. And we'll stay tuned.
Thank you very much, everyone.
Thank you. Bye-bye.